All right, welcome back, everyone. So this week we have a busy week. We're going to cover the rest of Chapter 9. So you had a little bit of an intro into cell division last week. So we're going to go through the cell cycle and mitosis and meiosis this week, and that will prepare us for our second midterm exam that occurs this weekend. So we're going to start out by looking at the cell cycle. So how cells, what they need to do in order to divide. So we'll discuss the events at interphase, and then M phase stands for meiosis or mitosis. And then we'll talk about the checkpoints that go into play in the cell cycle to prevent a cell from replicating when either it shouldn't be replicating or there's errors that have occurred. And then we'll talk about meiosis. So in meiosis, we'll look at the what the idea of haploid versus diploid, and why our gametes need to be haploid, whereas the rest of our cells are diploid, and we'll go into that. And then we'll talk about the stages of meiosis and some of the genetic variations that occur during meiosis that allow us to be genetically different from each other, which allows us to evolve. So we'll get into that today, or actually probably on Thursday. Well, we're going to focus in on Chapter 9 today, and then we'll do Chapter 10 on the next lecture. So in Chapter 9, again, we're going to take a closer look at cell division, which we started talking about last week, and then focus in on the cell cycle and molecular control systems. So this is a little bit of review. So we already talked about how life begets life. So we have this sequence of life stages leading from one adult to the next. In single-celled organisms, this involves reproducing the entire organism, right? So here's an example of a paramecium, not a paramecium, that looks like an amoeba dividing. So that single-celled protist will just get bigger, and then it just divides into two cells. Multicellular organisms, we use cell divisions in order to grow, renew, repair, and replace cells as needed. So we do this in order to replace the damaged cells throughout our body. So pretty much, whatever age you are, none of your cells in your body are the same age as you. Okay, you're constantly replacing them. Some of them might even be, an hour old. So we are multicellular organisms. We have to constantly be replacing the damaged cells and growing with new cells being added. Again, this is just a review, so I'm going through it pretty quickly. So we have, as eukaryotes, we carry our DNA on what we call linear chromosomes. So we actually have multiple chromosomes, and they carry different units of DNA, and our DNA carries the genes that make us. So we need a way that we can take all of our chromosomes, our 46 chromosomes, with all of our DNA, our entire blueprint to make another one of us. We need to find a way to duplicate those chromosomes so that when our cells do divide, that the DNA can go into the daughter cells and have exact copies. So all of our DNA is what we call the genome. So you have your particular genome that's housed on all of your DNA. Again, with eukaryotes, we carry ours on these many chromosomes, and it varies depending on the species, the number of chromosomes. We have 46 for humans. And prokaryotes, they just have one singular DNA molecule. Let's see. So there's our 46 chromosomes. Our chromosomes, as I stated before, are composed of chromatin. So chromatin contains the DNA, the double helix, that nucleic acid we talked about way back in macromolecules, as well as these histones. So the histones are a type of protein, and that's going to allow the DNA to become compacted into these chromosomes. So the DNA is just this long string, this double helix. And in order to pack it into a, some tight space, like this little tiny nucleus, the DNA winds around the histones. So you can see how they're winding around the histones to compact them until we end up with a chromosome. Now, before cell division, that chromosome needs to undergo DNA replication to replicate itself, so that when cell division occurs, each of the daughter cells get a copy. And so here's one chromosome that has been replicated. So now you see two chromosomes here. And those two chromosomes are held together by something called a centromere. So a centromere holds the two chromosomes together. And since these are identical to each other, they're called sister chromatids. So we're going to take a closer look at these chromatids as they go through cell division, and pay close attention to the centromere. Okay. So here's our a cell before it's undergone DNA replication. So we have a single chromosome. There it's undergone DNA replication. So now we have a pair of chromosomes that are held together by the centromere. So we call those sister chromatids because they're identical. And then at the end of cell division, the chromosomes have separated, the sister chromatids have separated into different cells. And since they're identical to each other, now we have two daughter cells that are identical to the parent cell. And then we talked about the two different types of cells that are in your body. We have the somatic cells, and then we have the gametes. So the somatic cells contain two sets of chromosomes. So they're called diploid, or two sets, and we'll get into that term in a little bit. So diploid means two sets of chromosomes. And these are our non-reproductive cells. Gametes, they only have one set of chromosomes, so they're considered haploid, and those are our reproductive cells. So our somatic cells are a typical body cell, and in order to get more typical body cells, we need to make exact copies of them. So if we need to replace skin cells, we need to make more skin cells. We don't want to change it into an egg or a sperm, right? So our somatic cells will undergo mitosis because it basically will create another cell that's identical to the parent. And so we'll talk about the different stages of mitosis. Oops, let me go back. And then gametes, they undergo a different form of cell division to produce the gametes, and that's through meiosis. So meiosis is taking a diploid cell. We call those diploid cells germ cells. We'll get into that later. And instead of somatic, because they're reproductive cells. And then they undergo a process known as meiosis, which ends up creating these gametes that have half as many chromosomes as the parent cell. And so they're going to be haploid. And each one of the gametes are a shuffling of all of that genetic material, so they're going to be unique. So we'll talk about that as well. But let's get into something called the cell cycle. So the cell cycle is something that all cells will go through when they are going to undergo cell division. So this actually doesn't matter if it's mitosis or meiosis that we're talking about. They all go through this life cycle, each one of our cells in our body. So we're going to go through the different stages. Now, if you take a look at this figure here, we can divide the cell cycle into two main phases. We have interphase and mitotic phase, okay? And interphase is the non-dividing phase of the cell cycle. So non-dividing, whereas mitosis or the mitotic phase is when they're actually going to be preparing for division, they're dividing. 90% of the time, the cell is in interphase, the non-dividing phase of the cell cycle. Only about 10% of the time will it be in the mitotic phase. Okay, and then the mitotic phase ends with something called cytokinesis. So when you see this word cytokinesis, think cyto as cell or cytoplasm, and then kinesis is dividing. So this is when it's physically dividing, and that occurs at the end of the mitotic phase, when we end up with two separate cells. Okay, so let's take a closer look at the different phases. So we know we've divided the cell cycle into two main phases. We have the interphase, the non-dividing phase, and then mitosis, the cell division phase. But within interphase, we can actually further divide it into three stages. And so we call the first stage the G1 stage or phase, the second stage as the S phase, and then the third stage as the G2 stage. And so we'll go into, I think, a little bit more detail of what these stages mean. But for now, whenever you see the G stages, think growth. Okay, it actually stands for gap, but I like to think of them as the growth phases because this is where the cell is just basically growing. Okay, so you can see when the G1 phase is occurring right at the end of cytokinesis, so we have this newly divided cell, and so that cell is going to spend some time growing. Okay, so it needs to add more endoplasmic reticulum and mitochondrium and stuff like that. So during that phase, it's just going to simply be growing and getting ready. And then the S stage of the interphase stands for DNA synthesis stage. So think of it as synthesizing the DNA, otherwise known as replicating the DNA. So this is when our DNA is going to duplicate. So we're going to take each chromosome and we're going to make a copy of it. So instead of having one chromosome, we're going to end up with a sister chromatid. That's a pair that's held together by a centromere. So look at that centromere. We got one here. We still have one here. So this is still considered one chromosome, but it has two copies. They're sister chromatids. Okay, so this is the S stage when we're duplicating the DNA. So we're getting identical copies made. And then, hey, what's G stand for? Growth. So it needs to grow again. So that when the cell does divide, that, you know, each daughter cell has the necessary organelles in order to, be a functioning cell. So you got to make some more endoplasmic reticulum and Golgi apparatus and all that fun stuff. So that occurs during the G2 phase. All right. So first checkpoint question of the day. A researcher treats cells with a chemical that prevents DNA synthesis from starting. This treatment would trap the cells in which part of the cell cycle? Well, first off, would it be interphase or mitosis are we talking about here? So when does DNA synthesis occur? Does that occur during interphase or mitosis? So that's going to occur during interphase, specifically the S stage of interphase, right? So if there's no S stage, okay, it's going to prevent DNA synthesis from starting. Which stage is it trapping the cell in? Okay, so we have no S stage. So if there's no S stage, then we can't have a G2 stage, and we can't have mitosis. So it's basically stuck in the G1 stage. Okay, so we're stuck in the G1 stage because we can't do the next stage after that, or the phase. You can say stage or phase. Okay, all right. So let's go ahead and review the cell cycle, and it actually goes into a little bit more detail and prepares us for mitosis. So we're going to watch our lovely Amoeba Sisters videos to help us. And it also talks a little bit about cancer, so that's a good thing.
Have you ever been sitting in class and thought to yourself, I wonder what my skin cells are doing right now at this very moment? This kind of pondering may be unique to me, maybe, but wouldn't we at some point wonder what our cells are doing right now? Because if you remember, as part of the cell theory, you are made of cells. All living things are made of one or more cells. Many multicellular organisms like you have cells that work together, working together as part of a body tissue. Body tissues working together as part of an organ. Organs working together as part of an organ system. Your cells are specialized to work in these different levels of organization. You have skin cells, stomach cells, muscle cells, just to name a few, and their functions need to be regulated. These cells actually are regulated as part of something called the cell cycle, and that is going to relate to my question of, I wonder what my cells are doing right now. Cells themselves can grow in size. But let's put it in perspective now. A multicellular organism isn't growing because each individual cell is getting bigger. A multicellular organism itself grows by making more cells, by the cells making more cells by dividing. That's cell reproduction. One reason that you're bigger than you were when you were 5, unless you are 5, is because your cells have divided to make more cells. Mitosis and the cytokinesis that follows that splits the cytoplasm allows you to make new body cells. But you don't want that cell division happening all the time. Why? It is likely that you've heard the term cancer before. We have had family members that have battled cancer before. It is definitely a relevant topic for all of us. Cancer is in part due to cells that divide too frequently. The cells are not regulated, they are uncontrolled. Cancer cells can have other problems too. They might not be able to communicate with other healthy cells. They may not be able to carry out normal cell functions. They may not securely anchor themselves like other cells do, which can make them more likely to travel somewhere else. Some cancer cells have the ability to secrete their own growth hormone that makes blood vessels divert over to those cancer cells and supply the cancer cell with nutrients, which can take nutrients away from healthy cells. Why do cancer cells become this way? Well, there is a lot of research in this area. With some cancers, there may be genetic links, making some cells more susceptible to having problems. These genetic factors might run in families. Exposure to toxins, radiation, excessive exposure to UV light, all of these can be risk factors for some cells to become cancerous. The uncontrolled growth that cancer cells have gives rise to more cells like them, which can develop into a tumor. Some tumors stay put, but some do not. Now, the good news is that scientists continue to develop better treatments, which include destroying the cancer cells with radiation or medication, such as chemotherapy, which will target cells that divide frequently. Maybe someday you will be part of helping to meet the challenge of trying to eliminate cancer, because the fact remains that these cells are not participating in the cell cycle like they should. So, what is the cell cycle? The cell cycle is often represented as a pie chart like this. Cells are either in one of two different phases. A phase called interphase, where the cells themselves are growing, replicating their DNA, doing their cell functions, or they are in M phase, which includes mitosis and the actual splitting of the cytoplasm, cytokinesis. So this M phase is where cells actually divide to make more cells, but cells spend most of their time in interphase. So most of the time, they're not dividing. Now, depending on what kind of cell, it might do mitosis more or less often. For example, your hair follicle cells do mitosis frequently, which is why your hair can grow at the rate that it does. It's also why many cancer drugs may also target hair follicle cells, because many cancer drugs go after cells that do cell division frequently. It's a big deal for cells to hit this M phase. If a cell has an error, a harmful mutation, for example, you do not want it to divide, because then it will create another cell that has this same issue. That's where checkpoints come in handy. Along the cell cycle, there are checkpoints to check that the cell is growing well and replicating its DNA correctly and doing everything it's supposed to do correctly before it divides. To better understand those checkpoints, let's further divide this cell cycle pie chart. We have G1, S, G2. All three of those are part of interphase. Then we have M phase, where mitosis will happen. During G1, the cell individually itself grows. Then it replicates its DNA in S phase. You can remember that because the S is for synthesis, which means to make something, and it's making DNA. Then G2, the cell grows some more in preparation for mitosis. So let's take a look at checkpoints. We've got one here in G1. This checkpoint checks, is the cell growing well enough? Is its DNA damaged? Because if it is, you definitely don't want it to move on to S phase, where it would replicate DNA. Does the cell have the resources it needs if it were to keep moving on? This checkpoint in G2 checks if the DNA was replicated correctly back in S phase. Is it growing well enough? Does it have the resources it needs to continue? Okay then, moving on. This next checkpoint in M phase is my favorite checkpoint. It checks in the stage metaphase to make sure the chromosomes, which are made of DNA, are lined up in the middle correctly, that they're all attached to the spindle correctly, because if they're not, The chromosomes will not be separated correctly. So now you may have two big questions. First, what happens if the cell doesn't meet the requirements of the checkpoint? And second, what is doing the regulating of this cycle anyway? To address the first question, if the reason the cell can't go past the checkpoint is a reason that can be fixed, the cell may kind of pause here until it can fix that issue. But if it can't be fixed, then the cell does something called apoptosis, which basically means the cell self-destructs. This ensures that a cell that is damaged beyond repair will not go on to divide. So what is doing the regulating anyway? We've mentioned before that proteins are a big deal. Genes in your body can code for proteins that do an assortment of functions, and there are many proteins involved with regulating the cell cycle. Some of them are positive regulators because they allow moving forward in the cycle, and some are negative regulators that might make things stop. The proteins themselves can be sensitive to cues inside and outside of the cell. So, for example, two proteins that are involved in positive regulation are cyclin and CDK. CDK is specifically an enzyme protein, a fancy kind called a kinase, which is worth a Google. CDK can have different forms of cyclin protein bound to it. Different types of cyclin rise and fall throughout the cell cycle, and the rising and falling is based on a variety of signals to determine when the cell should move on to the next cell cycle phase. Typically, each cell cycle phase, G1, S, G2, M, will tend to have a different cyclin binding with the CDK. The rise and fall of cyclin types and the role CDK has when it's active is a fascinating subject to explore. Remember that vocabulary word we said, apoptosis? Proteins that are negative regulators, for example, a protein called P53, can be involved in initiating apoptosis. Again, we encourage you to explore beyond the video. One last thing to mention. There are some cells that don't go through the phases we mentioned because they're actually in G0. That's a 0, by the way, and not an O, because if it was an O, then it'd say go, and G0 is kind of the opposite of that. G0 is a resting phase. Now, cells here are still performing cell functions, but they're not preparing to divide. Some cells go here temporarily, maybe if there's not enough resources around, for example. But some, like many types of neurons in your brain and spinal cord, may stay here permanently. If they stay here permanently, they'll never get to end phase, so they will not divide. This can be one reason why a major injury to the brain or spinal cord can have challenges with healing, as many of those cells may not be able to replicate, a topic that definitely continues to be researched. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay, so that was a good intro into the cell cycle, as well as you learned a little bit about cancer. And so with the cancer talk, basically what is cancer? It's just uncontrolled cell division within your body. And so you saw that there were several different ways to prevent it with those checkpoint systems that are in place during the cell cycle. So we had that gap, the one during G1, and then we had another checkpoint at G2, and then we had one in the middle of mitosis. And so those are going to hopefully ensure that the cell is dividing correctly. But sometimes we have those errors that occur where a cell will blow through the checkpoint, even though it probably shouldn't, and then start dividing when it wasn't supposed to. So we're going to now go into the dividing phase of the cell cycle. So we talked a little bit about interphase. Now we're going to get into the dividing phase, and we're going to talk about somatic cells. So remember, the somatic cells are your typical body cell. So think of your skin cell, your heart cell, any cell in your body, with the exceptions of the ones located in your ovaries or testes. So those somatic cells are going to undergo mitosis, and your book divides mitosis into 5 stages. If you've taken biology before, you may have seen mitosis in 4 stages. I learned it in 4 stages, but your book wants it to be in 5 stages. So we're going to learn it in 5 stages. You can tell I don't really like that. But anyway, we'll deal with it. So we're going to divide it into 5 stages. It bothers me that they have it in 5 stages. Normally, I guess I shouldn't say normally because your book is normal. In the stages that I learned it and the Amoeba sisters learned it, prophase and prometaphase are 1 stage. So if you learned it previously with 4 stages, just realize that your book has divided prophase into 2 stages. So you have prophase and prometaphase. Okay, it's fine. We'll work on it. And then we have metaphase, anaphase, and telophase. So we're going to learn about each one of these stages. While we're looking at it, we want to pay close attention to exactly what is happening to the chromosomes. So remember, the chromosomes before cell division, they've undergone DNA replication and they have those little sister chromatids. So we need to pay close attention to those sister chromatids, chromosomes throughout the mitotic process. Okay. And then at the end of cell division, in telophase, is when we have something called cytokinesis occur. So cytokinesis is basically when the cell physically divides into 2. So we're going to start out watching an Amoeba Sisters video to kind of go over the different stages. The Amoeba Sisters video goes with the 4-stage method, not the 5-stage method. So just remember, when they talk about prophase, your book divides prophase into 2 phases. Okay, so let's watch the video.
Have you ever looked down at your leg or your arm to find some cut and you have no idea where it came from or how you got it? So you put a bandage on it and a few days later it's gone. It's all healed and you don't even think anything of it, of this amazing process that causes this to happen. Or let's say you're looking at your nails and you notice they're a lot longer since the last time you cut them. Or let's say you're looking in the mirror and you notice that you are a lot bigger than you were when you were 5 years old. What do all of these things have in common? Well, one major thing they have in common is mitosis. Mitosis is a type of cell division done by most of your body cells, and it's really important for cells to divide. If they didn't divide, you wouldn't grow. I mean, how do you grow if you can't make more cells, right? So one reason why you're bigger than you were when you were 5 is mitosis. Mitosis also is great for repair of damage. If you have some kind of accident, like when we were talking about that cut on your arm or leg, well, you want to make sure it gets repaired well. So you have to make more cells to do that. Mitosis is great for that. Now, it's really important to understand what it is not. Mitosis is not a process that makes sperm or egg cells, because that's something different called meiosis, which sounds like mitosis, unfortunately, but it's a different process. Mitosis is done to produce body cells. Mitosis makes identical cells. That is the goal, identical cells. So if you're trying to make more skin cells to replace worn out or damaged skin cells, you don't want to start suddenly making stomach cells there. That would be ridiculous. You want to make sure you have identical cells replacing what was lost. So mitosis makes identical cells. It's a really important thing. Now, it's also important to understand that your cells are not dividing all the time. If all they did was divide, it would just be rapid, crazy growth. In fact, this is kind of what cancer is. Cancer is uncontrolled cell growth. We have a clip on the cell cycle and what the cell is usually doing most of its daily life, which is actually a phase called interphase, where it's growing and replicating its DNA and carrying out its daily cell functions. That's where cells spend most of their time in respect to the whole cell cycle. Mitosis is a very short amount of time in respect to the whole cell cycle, but mitosis is a critical process because this is where it is going to divide and make more cells. Before we get into the steps of division, it's really important to understand that your cells have something inside them, an organelle called the nucleus. And the nucleus holds your DNA. DNA is really important because it's your genetic information. And if you're going to make more cells, you need to have the same DNA in those new cells as you did in your original cells. You want it to be identical, no mistakes. Very important. The problem is you've got a lot of DNA, and we've got to get that DNA into the new cells using mitosis. So there has to be a better way to organize that DNA. Well, what actually happens is that DNA can be organized into these condensed units called chromosomes. Chromosomes are made of DNA and protein. You've probably heard before that humans have 46 chromosomes. That means 46 chromosomes are found in most human body cell nuclei. What are nuclei? Well, it's the plural of nucleus. You don't say nucleuses, you say nuclei. Well, in the nuclei, there are 46 chromosomes. Organizing DNA into condensed chromosomes makes it a lot easier to move over when you're making new cells. So if you have 46 chromosomes in a human body cell, you have to duplicate those chromosomes in interphase before mitosis starts. That basically means you are duplicating your DNA, since chromosomes are made of DNA and protein. You have to do this before mitosis starts because if you're going to make an identical cell that has 46 chromosomes, just like the original, well, it makes sense you have to duplicate the genetic material before splitting. So if you look at our cell cycle video clip, we talk about interphase. That's a stage where most of the time cells are spending their time. They're actually duplicating their DNA during that time. So ready for the tricky part? Well, because we tend to count chromosomes by the number of centromeres present, when the 46 chromosomes duplicate, we still say there are 46 chromosomes, as the sister chromatids are still attached, and we're counting by centromeres. So 46 chromosomes here, they replicate in interphase, and you still have 46 chromosomes in this picture, but you went from 46 to 92 chromatids. We have a video explaining that in more depth and how that factors in for mitosis. Okay, so now we can get right into mitosis. I like to tell students to remember PMAT. It's a little acronym that helps you remember. The P is for prophase, the M is for metaphase, the A is for anaphase, and the T is for telophase. So remember PMAT, the stages in order. The very first step is prophase. Prophase because it's the beginning step, the nucleus is still there. And it's going to go away later on, but this is the stage where it's actually still there. The chromosomes are visible. In fact, we say they're condensing, which means they are thickening and visible. The next stage is metaphase. M for metaphase, but I also like to remember M for middle because in this stage, the chromosomes line up in the middle of the cell. The nucleus has been disassembled, it's no longer there, so we've got the chromosomes in the middle waiting there. Next, the A is for anaphase. In anaphase, I like to think as the A for away. The chromosomes move away. They are moving to opposite sides of the cell, so they are moving towards the poles of the cell. Now, one thing to point out, these chromosomes, they're not moving by themselves. They actually have something called spindles. These spindles are fibers that help move the chromosomes to the ends. Kind of helps them move along. The last stage of mitosis, think T for telophase. In telophase, the chromosomes are actually at the complete opposite ends, and new nuclei are forming on each side to make these two new cells. The nuclei are starting to surround the chromosomes on both sides. I like to think of the T for 2 because you can really see in this step that the end goal is going to be 2 cells. And in the human body, they're each going to have 46 chromosomes. And again, remember, they are identical. Now, cytokinesis is responsible for the final separation into 2 cells by splitting the cytoplasm, which completes after the PMAT mitosis stages. So why did all this matter? Without understanding cell division, we wouldn't understand how growth and repair happens, because they both require more cells to be made. Understanding mitosis is also very important for cancer research. Cancer itself is uncontrolled cell growth. So in other words, uncontrolled mitosis. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay, so from that video, we basically got the different stages. And again, remember they have that acronym PMAT, which if you want to use the textbooks version with prophase and prometaphase, just add an extra P on there. So PPMAT. But we're going to go through each stage in detail. Now, as we go through each stage, remember the cell does not have specific stages. Okay, so what I mean by that is we're naming these stages based on what we see under a microscope. So the cell itself is in constant movement. There's no like stopping and saying, okay, well, I'm done with prophase. Now I'm going to get ready to get into metaphase. Nope, it's just a constant thing. So that's why there is some overlap with our textbook versus other textbooks with the number of phases. And also you saw from that video, it talked about cytokinesis taking place at the very end or outside of cell division. Well, it just kind of depends on your interpretation because the cell doesn't know, that it's done with telophase. It just starts dividing. So we're going to actually see a cell undergo mitosis, and I'm going to show you this video twice. I'll show you before we talk about the different stages in detail and then after. So right now you might not be able to identify when it has gone through a particular phase, but it's kind of cool to watch anyway. So we're going to focus in on this animal cell and pay close attention to the chromosomes. You'll see them. They look like little lines. Okay, so this is prophase. So you can see the chromosomes are condensing. And then it's starting, it's probably prometaphase at this point. And now they're going to be moving towards the middle. So that's middle metaphase. So they're still getting ready to move to the middle. So you can see this is the entire cell, by the way. So now it's in the middle. They're all lining up in middle metaphase. And then there's anaphase. You can see the chromatids moving away from each other. And then telophase, where they're dividing into two, ending with cytokinesis. Pretty cool, huh? Okay. So we're going to go through each individual phase in detail. And as we go through, let me just double check something. Okay, so as we go through, a lot of us are visual learners, so it would be very beneficial to the majority of you to draw pictures of each individual stage, okay? So that you can specifically see what is happening during each stage of mitosis, okay? So I'm going to start out by going through interphase, okay? So remember in interphase, during the G1 stage is the growth stage after mitotic division. And then we have the S stage, the synthesis of DNA stage. So in the S stage, we have our 46 chromosomes for humans, but there's 92 chromatids. Okay, so remember each chromosome at this stage contains 2 sister chromatids. So that once we're in G2, we're ready to divide. So let's take a look at that cell. This is a very simplified view of the cell in right before mitosis. So we're in G2 phase of interphase. So you can see this is the cell membrane. Just to give you some bearing here, we've removed all the organelles, so we just have the cell membrane. We have the centrioles. Hey, remember those centrioles? That's we have a centriole. We have paracentrioles, which our centrioles look a little different from plant cells. They just have one instead of a pair. So these centrioles, also known as centrosomes, I like to call them centrosomes because centrosome is alone in the cytoplasm, whereas centromere is in the middle of a sister chromatid. Might make you help you remember. So centrosome, think alone. Centromere in the middle of the chromosomes. Okay. So we have these centriole pairs or centrosomes in the cytoplasm. And then here's our nucleus, because this is a eukaryotic cell and only eukaryotic cells undergo mitosis. So we have in the nucleus, you see the little nucleolus. What does that thing do? Remember, it produces the components of ribosomes. And we have the nuclear envelope, with those little nuclear pores, and then we have our DNA. And at this point, look at that DNA. Can you identify individual chromosomes at this point? Nope. So it just looks like spaghetti string under the microscope. You can't really tell anything, okay? Now we're going to start mitosis. So we start with, remember the PP mat? So we have prophase, prometaphase, and then mat. So prophase, I like to think of the preparing to divide phase. So they, in the Amoeba Sisters video, they said pro means before. I think of it as preparing to divide, so P for preparing. And so there's a number of things that are going to occur to prepare for the division. And you notice the cells spent the majority of the mitosis time in that prophase or prometaphase, these two phases combined. So let's start out by looking at some things that are occurring compared to our interphase. For one thing, you see that these centrosomes, they're starting to move apart. So that's one thing. The centrosomes are going to move to opposite poles. So you can see them moving to opposite poles. By the end of prometaphase, they are now at opposite sides of the cell. Okay. Another thing you'll notice is that these centrosomes are producing mitotic spindle. So mitotic spindle, part of the cytoskeleton, is being produced by these centrosomes. Okay, so that'll become important because eventually they're going to attach to those chromosomes. Inside the nucleus, there's also some strange things occurring. Where'd the nucleolus go? So the nucleolus disappears. Also, look at those chromosomes. They have condensed, okay? So they're no longer these spaghetti strings, they have condensed. And you can actually physically start seeing them under a microscope. So you can see them condensing here, and then look at how condensed they are in prometaphase. In this image, you can see that they are definitely chromosomes at that point, okay? And at this point, each chromosome, remember, contains 2 sister chromatids, okay? Because we're counting the chromosome by the centromere. So there's the middle, the mere centromere. Okay? So in prophase, you can just define it as the chromosomes condense and the mitotic spindle starts to form. And then prometaphase, to separate that, you can think of the nuclear envelope disappears. So notice it's intact here, and so it's going to start to dissolve. And as it's dissolving, that allows the mitotic spindle to attach to the centromeres of the chromosomes. Okay, so you can see that mitotic spindle extending from the centrosomes. Remember, centrosome alone in the cytoplasm. And it's going to attach to the centromere middle of chromosome. And there's a special site, or basically it's a clump of protein on that centromere, which we call the kinetochore. And that's where the mitotic spindle attaches. Okay, and that mitotic spindle, as I said before, is part of the cytoskeleton. And you might recall from the cytoskeleton, there's 3 different types of cytoskeleton. The mitotic spindle is part of the microtubules. Okay. So there's our mitotic spindle. So by the end of prometaphase, it's ready to divide, okay? So it's prepared. We have the nuclear envelopes disappeared. The mitotic spindle has attached to the centromeres of the sister chromatids, and the centrosomes have migrated to opposite poles. So we are ready to go. Next, we go into where we do PP mat. So now we're in the mat part, metaphase. Like the video, I call it middle metaphase because that's when those mitotic spindle are going to push and pull and push and pull until the sister chromatids are aligned at the equator of the cell. So if they're at the equator of the cell, they're in the middle of the cell, okay? And those sister chromatids are being pushed and pulled by that mitotic spindle that's being controlled by the centrosomes. Okay, so once they're aligned in the middle, you see that they're ready to go. Okay, so metaphase, think middle metaphase. They're aligned at the middle. And then in anaphase, I like to think away anaphase. So this is where the centromeres dissolve. And now, if the centromeres are dissolving, what happens? They're no longer held together. The sister chromatids are no longer held together. And if they're no longer held together, then they are going to be pulled to opposite poles. Okay, so here we have away anaphase because the sister chromatids are no longer held together, and so they are separating and being pulled to opposite poles. So think anaphase away. And then finally, telophase, think telophase 2. Telophase 2, because they're going to divide into 2. And during this phase, I like to think of it as the opposite of prophase, because it's basically they're doing everything in reverse. So remember in prophase, we have the centrosomes moving to opposite poles, and producing mitotic spindle. Well, the mitotic spindle's going away, it's dissolving, there's nothing left, and there's, nothing left to go. The nuclear envelope was disappearing in prophase while it's reappearing in telophase. The chromosomes are condensing in prophase while they're going to be kind of unwinding it in telophase. The nucleolus disappeared in prophase while it reforms in telophase. So basically everything that happened in prophase, imagine the opposite occurring in telophase. And then while that's occurring, you start seeing the nuclei form. So we have this one cell here that actually has 2 nuclei at one point. And then you also start seeing it starts to pinch inward on its cell membrane or plasma membrane. So this pinching is called the cleavage furrow. So you start seeing a cleavage furrow form. This is for an animal cell. It's a little different for plant cells. But you start seeing this cleavage furrow forming. And this is where the cell kind of pinches inward and starts to physically divide into 2. So that's what cytokinesis is. Okay, so once cytokinesis is done, we end up with 2 separate cells. Each cell has its own nucleus with its own set of chromosomes. Okay. All right, so let's watch a video put apart by your textbook manufacturer that kind of goes through everything.
This soccer player's knee will be as good as new in a few weeks, thanks to mitosis, a type of cell division that generates new cells for growth and repair. Let's move into a cell to witness the events of mitosis. Before a cell can divide, it must first duplicate the chromosomes stored in its nucleus. During chromosome duplication, several bubbles open up along the chromosome. Each bubble grows until it merges with an adjacent bubble. Each chromosome now consists of 2 identical copies called sister chromatids. Getting closer, we see that each sister chromatid consists of DNA wound around small proteins called histones. The sister chromatids begin to coil into tight helical fibers. Outside the nucleus, centrosomes that duplicated earlier move away from each other to opposite sides of the cell. Microtubules extend from the centrosomes, forming the mitotic spindle. Back in the nucleus, the DNA forms loops, becoming more compacted. These structures fold back on themselves, eventually condensing into a shorter and thicker chromosome consisting of 2 sister chromatids. As the chromosomes continue to condense, the nuclear envelope breaks up. The array of spindle microtubules is now extensive, and the chromosomes are fully condensed. Spindle fibers from each pole attach to protein structures located at the centromere of each sister chromatid. As the chromosomes are bound by spindle fibers from opposite poles, they move first one way and then another. The counteracting forces of the spindle eventually cause all the chromosomes to end up at the center of the cell, as if arranged on an imaginary plate. The sister chromatids are released from each other, each becoming a full-fledged chromosome. They are moved toward opposite poles of the cell, pulled along the spindle fibers attached to them. At the same time, overlapping spindle fibers that are not attached to chromosomes continue to lengthen, pushing the poles farther apart. Once the chromosomes arrive at their destination, they become less condensed. Two new nuclear envelopes form, completing mitosis, the division of one nucleus into two genetically identical daughter nuclei. The cytoplasm divides by the process of cytokinesis, forming two separate daughter cells. In your body, millions of cells divide every second, providing new cells for growth and for repair of damaged cells.
Okay. So again, that video kind of showed you all of the different components of mitosis, and you even saw a little pro-metaphase because it was your textbook. But you could see exactly what was happening with the chromosomes. And so that's what's the most important part when you're talking about mitosis and meiosis, is what's going on with those chromosomes? How are they moving? So pay close attention to the chromosomes. So let's go through and just do some definitions, which we didn't define certain things when we were looking at the figures. So we talked about in prophase how the mitotic spindle begins to form. Well, what is mitotic spindle? So mitotic spindle, again, is part of the cytoskeleton, and it's made of microtubules as well as some proteins. And you saw that the proteins, they're going to help assist with the movement. So chromosomes are controlled by that mitotic spindle. The pushing and pulling of that mitotic spindle is telling the chromosomes where to go. And that's formed, the mitotic spindle is formed by the centrosome. So that's a type of microtubule organizing center. So that's replicated during interphase. I believe, I can't remember when in interphase, but I think it's during the it's probably during G2 actually. So it's probably during the G2 phase. So the centrosomes are replicated during interphase. And so now we have two sets of them, so that during mitotic division, during prophase, they can move to opposite poles. And they're going to produce the mitotic spindle. So the mitotic spindle kind of extends from the centrosomes in what we call an aster. So think aster as a star, aster star. So it does look like an aster as you know, these mitotic spindles are flowing away from the centrosomes. So we have these asters or star-like things with the microtubules extending from the centrosomes. All right. And then we have this mitotic spindle starts to attach to the chromosomes. And there's that special place on the chromosomes in the centromere region called the kinetochore. So there's that term kinetochore. So the kinetochore is just a group of proteins that the mitotic spindle attaches to. Okay, so the mitotic spindle tubules, microtubules attach to the kinetochores. Okay. And then that's what is going to be, that attachment is going to be pushing and pulling the sister chromatids or the chromosomes where they need to go. So at metaphase, the centromeres are going to be at the middle of the cell. So we call the middle of the cell the equator, also known as the metaphase plate. Okay. And then during anaphase, we end up having the kinetochore where the microtubules are attached. They're going to stay intact. The centromere just kind of dissolves. And then that's going to allow for the sister chromatids to pull away from each other by that mitotic spindle during anaphase. So anaphase away, the sister chromatids are being pulled away from each other. So here's a visual form. Here we have middle metaphase because they're lined up in the middle. You can see we have the centrosomes on opposite poles. There's the aster, which is basically that star-like thing where the mitotic spindle is extending from. You see the mitotic spindle is attached to a specialized protein complex called the kinetochore. So here's just a kind of a view of it. So that's the kinetochore. So that's where the mitotic spindle is attached. And notice each sister chromatid has their own kinetochore. Okay, so the mitotic spindle is not attaching to the entire chromosome, it's just attaching to an individual region of the sister chromatid. So that way, when we have anaphase, okay, you can see how that mitotic spindle from this particular sister chromatid is going to start to shorten. And as it's shortening, it's going to pull that sister chromatid away from its sister. Okay. And then finally, once we are done, we've undergone anaphase, completed that. We're in telophase. We've reformed our nucleus and everything. Then we need to start to physically divide into two. So that's called cytokinesis. And like I said, in animal cells, there's a formation of a cleavage furrow. So you can see here we have basically one cell with two nuclei at that point. And it's going to start pinching inward. And as it's pinching inward, plasma membranes form, separating into two cells. Okay, so that's called the cleavage or cleavage furrow. Plant cells, they're a little different because they have a cell wall. Okay, so how does that occur? So here is a plant cell that has basically undergone mitosis, and we have two nuclei. So right now we have a cell with two nuclei. So we got to complete cytokinesis. And since they have a cell wall, it's a little bit more complicated. So they have to form cell plates, which are the cell walls. So to do that, the cell plate actually forms in the middle of the cell. Okay, so here's this one giant cell that's undergone mitosis, and you're going to start seeing little cell wall plates forming. And those cell wall plates will extend until eventually you have a separation. Okay, so you can see the new cell wall that formally divides the two. Okay, so that's why it's a little different with animal cells and plant cells. So plant cells, think pinching inward. Plant cells going outward. It's going the opposite. It starts inward and it's moving outward. Okay. And then here we have our microscopic slides. And we'll actually look at cells under the microscope next week to demonstrate this. But you can see here's what a cell looks like in prophase. So you have the nucleus is disappearing. It's still kind of present. You can't see individual chromosomes, but they are condensing, okay, and there is a nucleus present. And then prometaphase, you can see the chromosomes are now fully condensed, so you can see the chromosome, individual chromosomes. And then here we have middle metaphase, so they're all lined up in the middle, pretty easy to distinguish that phase from prophase. And then anaphase, you can see that they've pulled away from each other. So that one is pretty easy to distinguish under a microscope. And then this one is in telophase, and you can tell that it's in telophase because I still see some gaps here. Okay, so I can see that there's a cell plate forming, but it's not fully formed. So what type of cell is this that's dividing? Is it an animal cell or a plant cell? Well, it's got to be a plant cell because of that cell plate. Plus, they look like little rectangles, right? And that's due to the cell wall. But let's go back to our animal mitosis video. So during this video, see if you can identify the different stages. Okay, so when is it completed a phase? When is it going into the next one? All right, so let's do that one more time and I'll walk you through it, okay? Let me just write something down. Okay, so we're in prophase. You still see the nucleus, but you see the chromosomes condensing. Here they're condensed, no more nucleus, so we're in prometaphase now. So in prometaphase, the spindle's kind of pushing and pulling them, and then we've moved into metaphase when they line up in the middle. So they're starting to get pushed and pulled towards the middle, so there's the metaphase plate. So this is my metaphase, and then you'll see them separate suddenly. There they go, that's anaphase because they're pulling away from each other. And then now they're in telophase, and you can see in telophase you start seeing the chromosomes unwind and you see cytokinesis occurring. Now if you pay close attention to the timer up here, from start to finish it takes about 2 hours for this particular cell. I don't know what type of cell it is, but it's not occurring like, in an instant. So when the video talked about how you make millions of cells every second, the process is longer than that. It's just you're constantly making new cells, but it takes a couple hours for it to occur. So that was mitosis. We've talked about how eukaryotic cells, they undergo mitosis, okay? Prokaryotic cells, they do not. They don't have a nucleus, so how can they go through mitosis? So they undergo something called binary fission instead. So binary fission is a very simplified type of cell division. So it's very simple. One slide will do it. So you can see here is our prokaryote. Remember, prokaryotes, very simple cells. They don't have a nucleus. They just basically have plasma membrane, ribosome, cytoplasm, cell wall. That's about it. They might have some other things in there, but no organelles. So we have, in this case, here's our single prokaryotic cell. Here is the DNA. Remember, the DNA is just one single strand, and it duplicates. Okay, so that's duplicating its DNA. And then it just divides. And that's it. Very, very simple. Okay, so there's no mitosis in prokaryotes. So we call it binary fission, where it's duplicating its DNA and then just simply dividing into two. So now we have two separate cells that are identical to each other. Okay, all right. Now, at the beginning of the lecture, we got into the cell cycle, and then we talked about mitosis. But during the Amoeba Sisters video, it talked about those checkpoints. So let's revisit that discussion with the checkpoints, and then we'll be done. So the molecular control system is basically a way that we can tell the cell to continue dividing if it's ready to go, or stop it when it shouldn't be dividing in the first place. So we have a couple of different, we have 3 different checkpoints that it goes through. So in order to continue, certain things need to happen. Okay, so if those things haven't occurred, then it stops the process, and then the cell either undergoes apoptosis, cell death, or it fixes whatever needs to be fixed. And then occasionally, if something goes through the checkpoint when it's not supposed to, we can end up with cancer cells. So that's uncontrolled cellular division. So the molecular control system failed somehow, and it allowed a cell to continue growing when it shouldn't. Okay. So let's talk about that cell cycle control system. And it is a sequence of events that occur, and it's regulated by both internal and external controls. So what I mean by that, internal controls versus external. So within the cell, there's certain things that can allow it to continue to go or stop. And then outside the cell itself, there could be like a hormone or something that prevents it from dividing or tell it to divide. So there's specific checkpoints that the cell cycle will stop until it receives a go signal. So there's one in the G1 phase, towards the end of it, one at the end of the G2 phase, and then one during the M phase, or metaphase part of mitosis. Okay. So let's start with the G1 checkpoint. So the G1 checkpoint basically decides if the cell actually needs to divide or not. So if it doesn't need to divide, it goes into the G0. So remember G0, that's not an O, that's a 0. So G0 means that it's basically out of the cell cycle. So sometimes, cells do this where they can be in the G0 phase for a year. So your liver cells, typically, they're not constantly dividing, so they'll be in the G0 phase for a year. Some of them might be a few days, like skin cells. They typically are replicating all the time because you're sloughing off skin cells all the time. Certain cells, like your neurons, they might spend years and maybe even most of your lifetime in the G0 phase. Okay, so that explains why, if you have a traumatic brain injury or you have your spinal cord injury, you're not going to regain that function because those cells don't replicate again. Okay, they're stuck in the G0 phase. So if you have damage to those cells, then unfortunately they stay damaged. There's nothing we can do about it. So take care of your neurons. But the G0 phase, that's basically the sleepy phase. The cell is doing its normal cell things. It's not going to actually go into cell division. But if the signal says, you know what, we need another skin cell, let's go ahead and say yes, then it's past the G1 phase. So there's a go ahead signal that allows that cell to continue into the cell cycle. So what's after the G1 stage? The S stage. So that's where it's going to duplicate its DNA and so forth. Okay. So once it's ready to duplicate its DNA, okay, and it's going to need to grow a little bit more to prepare for cell division, there is another checkpoint. Okay. I'm not quite sure why. Seems like these, oh, okay, that's why, because this one would be up. So imagine this red flag right here. There wasn't a separate figure, that's why. But before it goes into mitosis, it hits the G2 checkpoint right at the end of G2. So that G2 checkpoint basically stops the cell and just kind of double checks that everything is in order, that the DNA has replicated correctly and everything, the cell has enough to divide. So it's got the centrosomes that have duplicated, it's got enough Golgi apparatus for 2 cells and so forth. So if that signal has passed, then it undergoes mitosis. And then finally, the third checkpoint occurs in the middle of mitosis in basically right during metaphase. So you can see that it's passed through prophase, it's passed through prometaphase, and then right at metaphase is when it stops. And that one is basically where it's going to double check and make sure that the mitotic spindle have attached to the chromosomes correctly. So we don't want a chromosome to not have mitotic spindle attached. So it's got to make sure that everybody's got their own mitotic spindle, everything is being pushed and pulled in the right direction in order to pass this checkpoint. So it's going to stop, double check, make sure everybody's got their mitotic spindle in place, and then receive the signal to go ahead. Okay, so there it went ahead, and it can go ahead and divide now. All right. So these signals are all internal signals. So we're having, this is occurring within the cell. External signals might be growth factors. So growth factors are basically proteins that are released by certain cells to stimulate other cells to grow. So here's an example. We might have human fibroblast cells stimulating division to form more platelets. Okay, so imagine you have, like you saw the soccer player with his cut on his knee. We need more platelets to clog that that blood vessel that's leaking. And so the cells near the area will release this growth factor PDGF, which will stimulate the division of these fibroblast cells in order to go ahead and stimulate the growth of new cells in that region to help heal that wound from that blood rupture. Okay. So here's an example of that. You can see here's some cells that have been added to a petri dish. And then basically they've transferred those fibroblast cells to two different growth medias, one that has the growth factor and one that doesn't. And you can see when there's no growth factor, nothing is going to happen. When there is growth factor, that's going to be an external signal to cause the cells to grow. Okay. There's other things besides growth factors that can cause a cell to divide from an external signal. One would be in density-dependent inhibition. So cells, they're not going to divide if there are a lot of cells around them, okay? Because there's no room for a new cell to form. So there's a particular cell signal that actually prevents cells from dividing when there's a crowded situation. So that's a density-dependent inhibition. So we're inhibiting the cell from dividing when it's very densely packed. So most animal cells exhibit this anchorage dependence, where it must be attached to some sort of substratum in order to divide. So that's another way that in order for cells to divide, they must be attached to something. So here's a couple of things that need to occur in order for the cell to divide. It cannot be crowded, and it needs to be anchored. Cancer cells are a little different because there's something that affects the cell itself, that abnormal cell, that cancer cell, where it ignores the density-dependent inhibition signal. And so it'll start dividing when there's no room, okay? And it ignores the signal that the cell needs to be attached to something. So that means it can start dividing wherever. It doesn't have to be attached to anything. So that's an error in cell division, where we have the cancer cells are dividing when all of these external signals tell them they're not supposed to. So here is, normal cells, they will divide if there is enough room. So this one has plenty of room, so they will divide, and it needs to be anchored to the surface, so it's allowed to divide. Cancer cells, they will stop dividing, or excuse me, they'll continue dividing even if there's no room. Okay, so you can see here's, there's no room here, and then the cells are dividing and dividing, and they're lumping on top of each other, and they're not attached to a substratum. So this is indicating that they are still dividing despite the density-dependent inhibition factor and the anchorage dependence. Okay. These normal cells, if you scrape some away, then they can, there's a little bit more room, so these guys can divide and fill in the space. Okay. So cancer cells, they are abnormal cells that are not responding to the signals like they're supposed to. They tend to divide regardless of a growth factor. Okay, so they'll divide it if there's none there. Normal cells, they can be converted to a cancerous cell through something called transformation. We end up with basically a clump of abnormal cells. And that clump of abnormal cells are what we call a tumor. Now, there's different types of tumors depending on what's going on. If that clump of abnormal cells stays in one place and it doesn't go anywhere, we call it a benign tumor. So some of you, you might have benign tumors. No need to worry because they're just going to sit there. And you just have a clump of cells that shouldn't be there, but they're not doing anything, so it's okay. If it's a malignant tumor, that means that the cells start to invade other areas, And they can actually enter the bloodstream, and those cells can be exported to other areas of your body and start growing in other areas. And so that's called a metastasis, when those exported cells start growing in other areas of your body, and then you have cancer. So malignant tumors, think of, those abnormal cells are spreading into other locations, and that's when that person, needs to undergo some medical help. Benign tumors, they're benign. They're just going to sit there. You might have some on your body or inside, and there's no real need to worry unless they start to metastasize. So when they start breaking off and exploring other areas of your body and spreading those abnormal cells, that's when you have cancer. Okay. So here's an example. Here we have a clump of abnormal cells growing inside this person's breast, and you can see that it's growing and it's invading other tissues. So this is a malignant tumor. And then notice it's going to start spreading into the blood vessels. So as it's spreading into the blood vessels, these abnormal cells are going to start getting carried through the body, through the circulatory system. And so that means that this person's cancer has metastasized, it's spread. Okay? And it can start growing in other areas of the body. And we always indicate what type of cancer it is by the location of the original tumor. So we have, these are abnormal breast cells growing in other areas of the body. So that would be breast cancer. Okay. All right. So let's go ahead and leave you with this picture. This is basically, you can take a look at all of these different cells. And you're going to, see if you can examine them and figure out what states or what stages of mitosis they are in. So let's start with this cell right here. What stage would that one be? Okay, so this one to me looks like it's in prophase. Okay, so you can see the chromosomes are condensing. Okay, what stage would this one be? That one's easy, right? That one's anaphase because they pulled away from each other. How about this one? This one looks like it's at the end of anaphase, so some might argue that it's anaphase, but look, what's that? The cell plate is forming. So because of the cell plate forming, this is not anaphase, this is telophase. Okay, it's physically dividing into two here. Let's see, what else do we have? What stage is this one? This is interphase. Okay, you can see the nucleolus and everything, but you can't identify any chromosomes. Let's see. How about, are there any in middle metaphase that you can see? I can't see any. Well, this one's probably in metaphase, but it's so small you can't really see it. Yep, so I don't see any in middle metaphase, but we got the other ones, so we're good. All right, so we'll go ahead and stop right here. And then this is the end of chapter 9, so we're going to switch over to chapter 10 for the next lecture.